Hello students, welcome to EPG Pathshala. I am Dr. Chirantan Rawal from Dr. APJ Abdul Kalam Government College, Silvassa, UT of Dadra and Nagar Haveli. Today we are going to discuss about a module malfunction in glycolysis, which is a part of a paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. Our target is to understand the concept of malfunction. We will also explore the biochemistry during the malfunction of glycolysis, and this is associated with the malfunction of the glycolysis. Malfunction means failure to function normally. Regulation of metabolic pathways is essential to maintain a steady state, a steady balance within the cell, that is homeostasis, which brings the harmony, controlling the flow of intermediates through pathways, conserving energy, preventing excess product being made and exhaustion of substrate or a substrate cycles. There are many ways in which metabolic regulations is carried out. A number of these processes are incorporated in metabolism. Enzymes play a huge role in regulation of metabolic pathways. Malfunction of a glycolysis leads to a inborn metabolic disorders. It is also called inborn errors of metabolism. Inborn errors of metabolism are a set of genetic diseases occurs due to malfunctioning of a specific metabolic pathways. Consequences of the malfunction: block of the metabolic pathways. It leads to the accumulation, building up of a precursors found before the biochemical fault, which leads to a toxic effect. Deficiency of the final product metabolites of the metabolic pathways leads to a fault in energy production. Or it may lead to the activation of alternative pathway if present available, with the creation of potentially toxic metabolites usually not found in organism. Coexistence of above described mechanism is possible. Generally, a metabolic disease involves more organs. It also shows systematic symptoms. We will understand malfunction in glycolysis under following headings, that is hexokinase malfunction, PFK malfunctions, pyruvate kinase malfunctions, aldolase malfunctions. Hexokinase is present in most of the tissues. It has a broad specificity. It acts on many sugars. It has a high affinity for glucose. It is inhibited by the glucose 6-phosphate. It has a low capacity for working for a glucose. So, it is inhibited by glucose 6-phosphate, that is G6P. Hexokinase deficiency results into the autosomal recessive diseases. It causes hemolytic anemia. In hexokinase deficiency will increase the affinity of a hemoglobin for oxygen. This results into the difficulty in releasing oxygen to the tissues. It may cause a tissue anoxia. ATP formation will stop and causes hemolytic anemia. The PFK deficiency, that is another, that is phosphofructokinase deficiency, results into the genetic disorders. It results into the type 7 glycogen storage diseases. You will get many ideas about the glycogen storage diseases in module 28 and 29 of the same paper. It is observed due to mutation in the gene of M subunit of the PFK enzyme. This gene, that is phosphofructokinase gene, helps in the synthesis of phosphofructokinase. Phosphofructokinase phosphorylates fructose 6-phosphate preceding to its cleavage into glyceraldehyde 3-phosphate, which then enter into the glycolysis. A pyruvate kinase deficiency is usually received inherited disorder. This enzyme is important for RBC, that is red blood cell. A lack of this enzyme would result in hemolytic anemia. Similarly, Torres disease is a phosphofructokinase deficiency in the metabolic pathway. It is a glycogen storage disease, that is type 7. There are 8 different types of glycogen storage diseases. Glycogen storage disease is results due to the deficiency of different enzymes. In type 7 deficiency, breakdown of glycogen to glucose, that is a glycolysis, for energy cannot take place during the exercise. So, if a person do exercise, there will be no release of energy. Increase the level of glucose 6-phosphate in muscles, which then results into the muscle pain, fatigue. Similarly, the Tourette disease, that is a deficiency of phosphofructokinase in muscles and erythrocyte. It is a genetic disorder. It is directly related to the impairment in the glycolysis. In this disease, patient is unable to break down glycogen in the muscle cells. Treatments includes the dietary treatment. It has a genetic studies for the diagnosis. The genetic studies is required, muscle biopsy or quantitative analysis of the PFK enzymes. So, its treatment generally includes the dietary treatments. So, this is a concept map of our lecture. We'll first briefly review the glycolysis process, discuss the malfunction in glycolysis. We'll try to explore the regulations and the diseases associated with the glycolysis. Malfunction means a failure to function normally. Glycolysis essentially means glucose plus lysis, that is a breakdown of glucose. Gluco means sweet, while lysis means breakdown. This is one of the most ancient pathway known to humankind. Malfunction of glycolysis leads to inborn metabolic disorders. It is also called inborn errors of metabolism. Inborn errors of metabolism are a set of genetic diseases occurs due to malfunction of a specific metabolic pathway. Metabolic pathways consist of a number of processes. It involves sequence of chemical reactions. This reaction occurs in a human body on products of dietary or endogenous origin with the aim of make it available or avoids their accumulation. Consequences of the malfunction or the block of the metabolic pathway. It results into the accumulation of precursors found before the biochemical fault, which leads to toxic effects. Deficiency of the final product's metabolite of the metabolic pathway. It leads to a fault in energy production. Activation of alternative pathways. At present, activation of alternative pathways with creation of potentially toxic metabolites usually not found in organism. Coexistence of above described mechanism is possible. Generally, a metabolic disease involves more organs. It also shows systematic symptoms. Glycolysis consists series of biochemical reaction through which one molecule of glucose is metabolized to give 2 moles of pyruvate and 2 ATP as a net energy yield. Preliminarily, glycolysis is a process that takes place in cytoplasm. Both aerobic and anaerobic organisms have this pathway for glucose metabolism. This process is regarded as anaerobic as oxygen is not required at any stage of glucose metabolism. However, even in anaerobic organism, glycolysis function in the same manner as presence of oxygen does not affect the reaction. The end product of glycolysis is pyruvate, which can further be metabolized in two ways: anaerobic, aerobic. In the latter half of the 19th century, a complete map of glycolysis was elucidated. And then Mayer of Parnas were the pioneer in the work, and hence the pathway is also known as Amden-Mayer of Parnas pathway. Similarly, many other scientists like Norberg, Warburg, Corey have significantly contributed in shaping the present knowledge of glycolysis and its related biosynthetic mechanism in a cell. Enter-Diodrop pathway is a name given to glycolysis reactions specifically when occurring in prokaryotes, archaea, after the contribution of Diodrop and enter. A metabolic disease occurs due to malfunction of a specific enzyme or of a deformity of a cellular transport mechanism. Glycolysis pathway has a total 10 reactions, at the end of which 2 moles of pyruvate is formed from single glucose molecule. There are many enzymes involved in this process. Certain reactions are reversible, while certain reactions are irreversible. Glucose to glucose 6-phosphate is a phosphorylation reaction require energy from ATP. The reaction is irreversible and it is catalyzed by the enzyme hexokinase. For your information, hexokinase is the enzyme that initiates glycolysis in muscle, brain and other vital tissue, while the glucokinase is the enzyme that does similar reaction but in liver and pancreas. Glucose 6-phosphate to fructose 6-phosphate is an isomerization reversible reaction catalyzed by glucose 6-phosphate isomerase. Fructose 6-phosphate to fructose 1,6-bisphosphate is another irreversible reaction of this pathway, which is catalyzed by PFK. Fructose 1,6-bisphosphate to 2 molecule of glyceraldehyde 3-phosphate. Fructose 1,6-bisphosphate actually splits into 2,3 carbon moiety of aldehyde and a ketone, which results into the formation of dihydroxyacetone phosphate. This reaction is catalyzed by fructose bisphosphate aldolase enzyme. 2 molecules of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, which is a dehydrogenation reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Here in this reaction, NADH is formed. Additionally, above oxidation reaction is coupled with the phosphorylation reaction that yields 1,3-bisphosphoglycerate. This eventually converted to 3-phosphoglycerate in an exergonic reaction, which is catalyzed by phosphoglycerate kinase. High energy bond in 1,3-bisphosphoglycerate is hydrolyzed to a carboxylic acid and the energy liberated in that is used conventionally to form energy-rich compound like ATP from ADP. 3-phosphoglycerate to 2-phosphoglycerate requires where phosphate group shifts from the third position to second position. The reaction is an isomerization reaction and is catalyzed by phosphoglycerate mutase. 2-phosphoglycerate to phosphoenol pyruvate, a dehydration reaction catalyzed by enolase. Phosphoenol pyruvate to pyruvate is an irreversible reaction which is catalyzed by transferase activity followed by dephosphorylation. There are certain malfunctions in glycolysis has been observed, mainly hexokinase malfunction, PFK malfunction, that is phosphofructokinase malfunction, Pyruvate kinase malfunction and aldolase malfunction. Hexokinase malfunction. Hexokinase is present in most of the tissues. It has broad specificity. It acts on many sugars. It has high affinity for glucose. It converts glucose into glucose 6-phosphate during the low blood glucose level. It has low Vmax, so it cannot phosphorylate high glucose quantity. It has low capacity for working on glucose. It is inhibited by glucose 6-phosphate. Hexokinase deficiency, it's a kind of deficiency of hexokinase which results into the autosomal recessive disease. It causes hemolytic anemia. In hexokinase deficiency, 2,3-diphosphoglycerate level is increased. This will increase the affinity of hemoglobin for oxygen. This results into the difficulty in releasing oxygen to the tissue. Finally, it causes the tissue anoxia. ATP formation will stop and causes hemolytic anemia. Phosphofructokinase malfunction. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate by phosphofructokinase. Low energy, that is high content of AMP, will activate the phosphofructokinase. ATP inhibits the phosphofructokinase. Phosphofructokinase deficiency results into the genetic disorder. It results into the type 7 glycogen storage disease. It is observed due to mutation in the gene of M subunit of the phosphofructokinase enzyme. It is designated as PFKM gene. This gene helps in the synthesis of phosphofructokinase. Phosphofructokinase phosphorylates fructose 6-phosphate preceding to its cleavage into glyceraldehyde 3-phosphate, which enters the energy generation phase of glycolysis. Glycogen storage disease, type 7 thyroid disease. It is a disease due to the deficiency of phosphofructokinase in muscle and erythrocyte. It is a genetic disorder. It is directly related to the impairment in glycolysis. In this disease, patient is unable to break down glycogen in muscle cells. It results into the interference in the functioning of muscle cells. Its diagnosis is done by genetic studies, muscle biopsy, or a quantitative analysis of phosphofructokinase enzyme. Its treatment includes the dietary treatment. Pyruvate is the end product of glycolysis. Two pyruvic acid molecules are formed from the two molecules of phosphoenol pyruvate. This reaction is catalyzed by pyruvate kinase. Pyruvate kinase deficiency is observed. The red blood cells do not have a mitochondria. They depend on the glycolysis. So, pyruvate kinase deficiency causes the low glycolysis within the cell of the RBC. It affects the sodium potassium pump, and it ultimately leads to the accumulation, the building up of intermediates of a glycolysis. The pyruvate kinase deficiency, the 2,3-DPG level is increased, which ultimately affects their affinity for hemoglobin. Finally, it causes the anoxia and leads to the anemia. It's a genetic disorder, obviously. It is an autosomal recessive disorder. It results due to the deficiency of aldolase enzyme. It causes due to the homozygous mutation in the Aldo-A gene. This Aldo-A gene encodes the fructose 1,6-biphosphate aldolase A, which catalyze the aldol reactions. A total 10 diseases are said to be associated with glycogen storage diseases. Pyruvate kinase malfunction. Pyruvate is the end product of glycolysis. Two pyruvic acid molecules are formed from the two molecules of phosphoenol pyruvate. This reaction is catalyzed by pyruvate kinase. Pyruvate kinase deficiency occurs. In red blood cell, there is no mitochondria. They are dependent on the glycolysis for their energy requirement. So, pyruvate kinase deficiency causes low RBC glycolysis. This low ATP level affects sodium potassium ATPase pump, and which leads to the damage to the membrane and lysis and hemolytic anemia. It leads to the abnormal accumulation of intermediates of glycolysis. In pyruvate kinase deficiency, 2,3-DPG level is increased. This will increase the affinity of hemoglobin for oxygen. Finally, it causes the tissue anoxia. ATP formation will stop and causes hemolytic anemia. Aldolase malfunction. Aldolase A malfunction is an genetic disorder. It is an autosomal recessive disorder. It results due to deficiency of aldolase A enzyme. It is caused due to homozygous mutation in the ALDO-A gene. ALDO-A gene encodes fructose 1,6-bisphosphate. Aldose-A catalyzes the aldol reaction. This reaction is irreversible to act on the fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is split into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glycogen storage disease type 12. This is also known as glycogenosis type 12, red cell aldolase deficiency or aldolase A deficiency. Again, it is a genetic disorder. It is autosomal recessive disorder. It results due to the deficiency of aldolase A enzyme. It caused due to the homozygous mutation. This is the same story that we learned in the previous slide. Aldolase A catalyzes the aldol reactions. Aldolase A is predominantly found in the skeletal muscles and the erythrocytes. There are certain reports that the mutation in the ALDO-A gene, specifically ASP128 while Kruder et al. In 1996 identified homozygous mutation in the ALDO-A gene, that is in glutamic acid 2 lysine. In this chapter, we learn about the malfunction of glycolysis occurs due to genetic alteration. Malfunction of glycolysis results into the genetic disorder. These unique diseases are quite varied in age of onset of symptoms, morbidity and mortality. These diseases are classified according to their individual enzyme deficiency. Each of these enzymes regulates glycolysis. So students, let us summarize what we learn in this module. This module deals with the malfunction in the glycolysis. Now you may be aware with what is malfunction. There are many enzymes that are involved in the glycolysis. If there is error related to this enzymes, it ultimately affects the reaction that they carry out. When there is a malfunction with the enzyme, malfunction with the glycolysis, it results into the abnormal diseases. We learn about the malfunction in glycolysis, but if we carry out the proper diagnosis and the treatment of the disease associated with the malfunction of glycolysis, then we may reduce the suffering to the patients. It is very important to do the early diagnosis and the treatment of a disease associated with this malfunction. It improves the quality of life. It reduces the damaging effect on the organs. Certainly, if there is an accumulation of any metabolites beyond certain limits, that will affect the concerned organ. But if we do a proper diagnosis and the early treatments will certainly reduce the pressure on the organs, which occurs due to the accumulation of metabolites, which ultimately extends the patient's lifespan. More recently, enzyme replacement and the gene therapy have been explored, but these techniques are under trial. It is not widely used at the patient level. In certain animal models, we have successfully used the enzyme replacement technique. We have used the gene therapy. You might be aware what is gene therapy. If you treat any disease by changing the gene, by incorporating a new gene within the system, then it is generally said to be a gene therapy. But gene therapy models have given a promising results in animal models, and skeletal muscle cells obtained from the affected proteins. In such cases also we got good results. In addition, adenovirus vectors for recombinant and transgenic enzymes to replace the defective or missing enzymes have shown a great promise in well-controlled laboratory studies. So ultimately these diseases cannot be cured by taking a simple medicine, because there is a malfunction within our system. It is there due to the defect in the enzymes, defect in the genes. That's why the enzymes cannot perform their task, and if enzymes are not able to perform their task, it leads to a problem, it leads to a disease. So certainly the cure of such a disease will be by doing an enzyme replacement or a gene therapy. We may, up to certain extent, can cure the patient from the symptoms. Now we do only the symptomatic relief to the patients. We treat the symptoms but not the disease. But by using such a technique like gene therapy and enzyme replacement, we may able to cure the patient in future. Thank you very much.